Okay, so welcome back to this next video in which we are discussing Cys-Loop receptors or Cys-Loop ligand-gated ion channels. Okay, right, so we've now discussed lots of examples of Cys-Loop receptors. What I want to turn my attention onto now is the structure of Cys-Loop receptors, and we're going to look at them in much more detail than we have previously. Okay, right. So firstly, what I want to talk about is how the structure of Cys-Loop receptors is split into three parts. Okay, so let me draw out a Cys-Loop receptor very basically again. Okay, so let's say this is going to represent the cell membrane here, the outer leaflet and the inner leaflet again, and now I'm going to draw my Cys-Loop receptor sitting in the cell membrane here. Okay. And it's going to have three separate domains: the extracellular domain, the transmembrane domain, and in the case of the mammalian Cys-Loop receptors, which are the ones that really we're the most interested in, okay, because they're the ones that are in humans, you're going to have an intracellular domain as well. But in the case of the non-mammalian ones, glucl, ilic, and glic, you don't have that intracellular domain. So let me now split it into these three different domains. Okay, so here's the extracellular domain here. Here in the middle is the transmembrane domain, and here is the intracellular domain. Now let me add on the pore down the middle there and split it into 5 separate subunits. So here is one of the subunits, and I'll draw a line down there. Okay, so that's all one subunit of our Cys-Loop ligand-gated ion channel. Here's another subunit here. Again, I'll draw a line down there, that's all one subunit. Okay, third subunit here, and then the fourth subunit there. Okay, right, so now let's highlight up these different portions. So I'll colour in this portion here in blue. These are the 5 extracellular domains of the Cys-Loop ligand-gated ion channel subunits, and together they all make the extracellular domain of the Cys-Loop ligand-gated ion channel pentamer. Okay, so this is what we'll abbreviate down to the ECD for extracellular domain. I suppose I probably should write it out in full at least once. So this stands for extra, that's the E, and then cellular, that's the C, and then D for domain. Okay, and that's the portion that's going to contain the Cys-Loop, although as we will see, it contains far more than just the Cys-Loop. It has, in fact, a beta sandwich that consists of 10 beta strands. Okay, and we'll discuss that more later on. Okay, then underneath the extracellular domain of each one of those 5 Cys-Loop ligand-gated ion channel subunits, you have the transmembrane domain. Okay, which is the portion that we will start off by discussing. This is the portion that actually forms the channel through the cell membrane. This is the bit that actually is sitting in the phospholipid bilayer here. Okay, so this is now what's known as the TMD. Okay, and that stands for transmembrane domain. Okay, so T for trans, M for membrane, and then D for domain. Okay, right. So we'll start off by discussing the transmembrane domain. Now the extracellular domain and the transmembrane domain are both conserved for all the mammalian Cys-Loop ligand-gated ion channels and all of the non-mammalian ones that I've discussed, the glucl channel from worms and ilic and glic from prokaryotes. Okay, the portion that I'm about to now tell you, okay, which is down here, and I'll colour in green, which is the intracellular domain, that is not there in the prokaryotic ones, that's only there in the mammalian ones. Okay, so this will be here in the nicotinic acetylcholine receptors, the GABA A receptors, the glycine receptors, and the 5-HT3 receptors, but not in glucl, ilic, and glic. Okay, so this final domain down here, this is what's called the ICD for intracellular domain. Okay, and I'll just underline that in green. Right, okay, so the way we're now going to go through discussing the structures of these different portions is we're going to start off by looking in detail at the transmembrane domain. Okay, and we've looked at the 4 membrane spanning alpha helices which make up the transmembrane domain for each of the 5 subunits of the Cys-Loop ligand-gated ion channel, but now we want to see how those 4 helices are going to be arranged to actually make the transmembrane domain. Okay, then what we'll do is we'll turn our attention onto the intracellular domain, and then finally we'll go to the extracellular domain. Okay, and then what we'll discuss is the actual gating mechanism by which, when ligand binds to the extracellular domain, it actually results in the opening of the channel. Okay, right then, let's start off with the transmembrane domain then. Okay, so I'm going to draw that basic picture that we had before of what a single subunit of a cis-loop ligand-gated ion channel looks like again, okay, so that I can color code those 4 membrane-spanning alpha helices. Okay, so once again, here is the cell membrane. Okay, then we have the N-terminus of our cis-loop ligand-gated ion channel subunit here. Okay, then we have that important structure, the cis-loop here, which is the structure in the extracellular domain after which the entire family of ligand-gated ion channels is named. Okay, then we have our transmembrane domain. Here's M1, M2, then we've got M3, and then the remaining ones will have this large loop between M3 and M4, and then a very small portion after M4 on the extracellular side, which then ends with the C-terminus. Okay, right. So let's now color code the 4 membrane-spanning alpha helices in, okay, so that I can now show you where they are in the actual transmembrane domain of the entire cis-loop ligand-gated ion channel, basically. So we've color coded M1 in blue there. Okay, we'll now have M2 in red. Okay, then we'll have M3 in yellow here, and I hope that shows up. Okay, then we'll have M4 in orange here. Okay, now what I want to do is go back to the picture of the transmembrane domain of the entire cis-loop ligand-gated ion channel and show you how these are arranged to basically produce that transmembrane domain. So basically what I'm going to do is I'm going to vanish away the extracellular domain, vanish away the transmembrane domain, and just now vanish away the intracellular domain, and just look at the transmembrane domain. Okay, so I'm going to cut these bits away, and now we're just going to have this transmembrane domain, which is this pentamer of these 5 transmembrane domains of the 5 cis-loop ligand-gated ion channel subunits in the cell membrane, and we're going to see how the membrane-spanning alpha helices of each of these 5 cis-loop ligand-gated ion channel subunits are arranged in that pentamer. Okay, right. So here now is the cell membrane zoomed up a little bit. Okay, and now here is just the transmembrane domain. So I've got rid of the extracellular domain and the intracellular domain, and we're now just looking at the transmembrane domain here. Okay, so it's sitting in the plasma membrane like so. Here is the pore going down the middle, and it's obviously going to be made up of these 5 separate subunits. So there's one, there's the second one, third one, fourth one, and fifth one. Okay, and I'll split them up down here, like so. Oops. Okay, right. Now what I want to do is colour in on the top of these where the positions of all 4 of these membrane-spanning alpha helices are for each of the 5 subunits. Okay, so each one of these 5 cis-loop ligand-gated ion channel subunits has the same basic structure. So all of them have 4 membrane-spanning alpha helices, and all of them have their M2 helix right at the centre here, lining the pore. Okay, so I'll put 5 red dots there. Okay, 1 representing each of the M2 helices for all of those 5 subunits of the cis-loop ligand-gated ion channel. Okay, so it's the M2 helices then that actually line the pore. Okay, that's a really important fact. M2 lines the pore in all cis-loop ligand-gated ion channels. And I should just stress, what we are now discussing, this holds for all of them basically. This isn't one specific one. Okay, this holds for all of them. Okay, so the M2 helix is the one which is lining the pore. So every single one of those cis-loop ligand-gated ion channel subunits contributes its M2 helix to produce this pore. Okay, right. Then the ones that sit behind the M2 helix are the ones that are next to it in the primary structure. So M1 and M3 sit behind it. Okay, so now I'll firstly put on M1s. Okay, so the M1s sit behind M2, and they sit to the left, basically, of M2. Okay, so over here. Okay, and I'll make that dot a little bit bigger. So there's our M1 for that one. There's our M1 for that cis-loop ligangated ion channel subunit. Here's the M2 for that one. Sorry, M1 rather. And there's the M1 for that one. Okay, so there are all the M1 membrane spanning alpha helices shown. Now let me add on M3 in yellow here. So M3 sits next to M1, okay, but to the right of it if we're looking from the front at this one here, okay. And then there's M3 for that one, there's M3 for that one, and there's M3 for that one. Okay, so behind M1, or peripheral to M1 if you like, you then have, behind M2, okay, I'm getting confused, start again. Behind M2 in this structure, which is the one that's in the center, you then have M1 and M3, which are the ones which are closest to it in this structure. Okay, so here is M1 in blue, so above M1 you'll then have the portion that makes the extracellular domain, which we'll discuss later on. Okay, then M1 spans the membrane, then you have the M1, M2 loop, then you'll go into M2, which is the one that's right in the center here, and which lines the pore, then you'll have the M2, M3 loop, and you'll go to M3 here, that will sit alongside M1, roughly at the same sort of peripheral level as M1, okay, it will then span back down, and now finally M4 is the one that's right out on its own at the outsides, basically. Okay, so it's the most peripheral. So here is M4 shown here. Okay, in orange. Okay, right, so that's how the 4 membrane-spanning alpha helices are arranged in each one of these cis-loop ligangated ion channel subunits, okay, in the cis-loop ligangated ion channel pentamer. Okay, right. What I now want to do is imagine that we can cut through the cis-loop ligangated ion channel right down the center, and I want to look at the cross-section that we produce, okay, because there's going to be a very important thing that we need to understand, which is this concept of the hydrophobic girdle, which narrows the pore right at the center. Okay, right. Now I also would just like to stress a point that I haven't stressed previously, okay, which is that this transmembrane domain actually goes on for about a nanometer above the plasma membrane on the extracellular side. Okay, so let me just label up this picture slightly better. So this is the extracellular fluid side, and this is the cytoplasmic side. Okay, now the thickness of the cell membrane is usually around 3 nanometers. Okay, and the transmembrane domain of your cis-loop ligangated ion channel will go through that entire 3 nanometers, but it also extends above the plasma membrane on the extracellular side by about 1 nanometer. Okay, right, so that's why I have it extending beyond the plasma membrane on the extracellular side, but not extending beyond the plasma membrane on the intracellular side. Okay, you will have the intracellular domain obviously on the intracellular side in the case of the mammalian ones, which we'll discuss later. Okay, but the transmembrane domain in all of them extends beyond the plasma membrane in on the extracellular side, basically. Okay, right, so now what I want to do then is cut through my cis-loop ligangated ion channel pentamer right down the center, and I want to have a look at the pore, basically. Okay, so what I'm going to now see is something like this. So here is the plasma membrane again. Okay, here is my transmembrane domain extending beyond the plasma membrane on the extracellular side. Now stress again, we are only showing the transmembrane domain of the cis-loop ligangated ion channel. Okay, we're not showing the full thing. Okay, like so. And basically it bends in towards the center, like so. So it gets to its narrowest point at the center. So that's one half of it. Now I'll show the other half of it here. Okay, like so, and again it bends in like so to make a narrowing right at the center of the plasma membrane, basically, like so. Okay, and this, what we're showing here, is the closed state of the channel. Okay, and it's this narrowing here that is keeping the channel closed. If that was released and these were moved back, then the channel would open. Okay, right, so what I'm showing you here is the closed channel. Okay, and this will be very important for our understanding of the gating mechanism later on. So basically, I've told you that the M2 helices line the pore. So basically, this is the M2 helix here, which is bent in this way inwards. So it bends inwards towards the center, has its narrowest point. The pore has its narrowest point right in the center. Okay, and then it opens back up towards the outside and opens back up towards the outside. This is the outside here, and opens back up towards the inside here as well. Okay, right, so what we now want to discuss is what holds the M2 helix kinked in this way, what holds it narrowed in the centre like that. It's almost as though there is a belt strapping the M2 helices together basically, and remember there will be 5 M2 helices. I've only got 2 shown here, but in reality there will be 5, okay, and this will be a 3-dimensional structure, okay. So it's almost as though the M2 helices are all sort of bunched together, held together by some sort of girdle, okay, something that holds them narrowed in at the centre there, okay, and indeed there is, okay, and this is what's known as the hydrophobic girdle, okay, this sort of concept of there being a belt that is holding all of the M2 helices very close together to narrow the pore at the centre there. Okay, so this is called the hydrophobic girdle. So what actually is then the hydrophobic girdle? What is it that is holding all of those M2 helices close together like that? Okay, well basically there is a ring of hydrophobic residues at this level. Okay, and what I mean by that is that every single one of the M2 helices at this level has a hydrophobic residue at that position facing into the pore. Okay, and what ends up happening is these hydrophobic residues all want to interact with one another. Okay, and the only way that they can interact with one another is if they get close enough to one another. Okay, and this is what drives all of these M2 helices to bunch together like this. Okay, so that all the hydrophobic residues can interact with one another. So at this level, the M2 helices have a lot of hydrophobic residues. These all want to interact with one another, okay, so that they can avoid water. And to do that, they have to bring the M2 helices close together. So all of the M2 helices end up invading into the pore. Okay, they head towards the central axis of the Cys-Loop ligangated ion channel. Okay, and that is what causes this narrowing of the pore at the center, because all of these M2 helices are sort of bunched up together so that their hydrophobic residues can interact with one another. Okay, so let me give you some examples of the hydrophobic residues that the M2 helices have at this level. Okay, so the main two are valine and leucine. So let me show you valine residues and leucine residues. Okay, right, so I'll start by showing a valine residue. Okay, so again, I'm drawing an amino acid residue, so I'll show the amino group here as though it's bound to the carboxylic acid group of the amino acid prior to it. Okay, and so far all of this is just the core amino acid structure. Here's the alpha carbon, here's the carboxylic acid group, again bound to the amino group of the next amino acid along. Okay, and the R group of valine then consists of a carbon with 2 methyl groups coming off it. Okay, 1 and 2 there. Okay, and then also a hydrogen coming off it here. Okay, so that's valine. Okay, and you can note that all of the structure, all of this R group here is very, very neutral. Okay, and that makes it very, very hydrophobic. It means that it doesn't interact well at all with polar water molecules. Okay, so that's a very hydrophobic side chain there. Okay. And then the next example is the amino acid leucine. Okay, leucine is basically just a bigger valine. Okay, so once again I'll just draw out the amino acid residue structure. So here's the amino group, the alpha carbon with the hydrogen coming off it, the carboxylic acid group, and now leucine is just valine but with an additional methylene group stuck in here. Okay, so there's that additional methylene group, and now the structure is exactly the same. So you have a carbon with 2 methyl groups coming off it, like so, and a hydrogen coming off it as well. So it's just a bigger valine, bigger and more hydrophobic than valine. Okay, so basically at this hydrophobic girdle level of the M2 alpha helices in all cis-loop ligand-gated ion channels. This is the thing, okay? Even though we've talked about a huge number of different cis-loop ligand-gated ion channels in a huge number of different forms of life, okay, and the subunits that make up these different forms of cis-loop ligand-gated ion channels will have very different primary structures of amino acids. At this mid portion of the M2 helices, they all have hydrophobic amino acids there, valine and leucine residues, okay? And so that these valine and leucine residues can interact with one another, the M2 helices end up sort of bunching together at the middle there, okay, as though there's some sort of string around them constricting them together there, okay, and that is what is known as the hydrophobic girdle. A girdle is just something that you can push around your waist or something, similar to a belt or something along those lines that can fin it up, basically. Okay, so that's why this is called a hydrophobic girdle, because it's narrowing the waist, if you like, of the M2 alpha helice bundles. Okay, right. So that's what keeps the channel closed, basically, this hydrophobic girdle, keeping the M2 helices close to one another. That's what keeps the channel closed. Okay, when you've got this narrowing right in the center, ions cannot move through it, whether this channel is conductive for sodium and potassium ions or chloride anions, okay, you can't get any ions through it whilst you've got the hydrophobic girdle assembled. So now, if you want to open the channel, what needs to happen is you somehow need to break these interactions between the hydrophobic amino acids at this level of the M2 alpha helices, okay? And if you can break those interactions, then you can imagine that the M2 helices will spring back outwards and open that pore up then now, okay, hugely reducing how narrow the pore is at the center there. Okay, and indeed we will see that the way that you open the channel is by breaking the interactions that hold this hydrophobic girdle in place. Okay, and we'll discuss that in more detail once we've done the extracellular domain and how ligand combines to the extracellular domain. Okay, right. So the next thing that I want to discuss with regards to the transmembrane domain is how selectivity is achieved. Okay, so this is how basically you gate the pore of the transmembrane domain. What I now want to talk about is selectivity. Now, what do I mean by that? So that sounds like one of these clever concepts, but it's a really important concept. Okay, selectivity just refers to the fact that some of our cis-loop ligand-gated ion channels select for sodium and potassium ions, the positively charged ions, the cations, okay, whilst other cis-loop ligand-gated ion channels that we have seen select for negatively charged chloride anions. Okay, what is the fundamental difference between the cis-loop ligand-gated ion channels that let positively charged ions, the cations, move through them, and the cis-loop ligand-gated ion channels that let chloride anions go through them? How does the transmembrane domain select for either cations or chloride anions? Well, I think the best way to talk about this, rather than trying to do it in general, is to just discuss an example, and then after we've got the example, I will then go back and generalize it for all cis-loop ligand-gated ion channels. So I'm going to do an example basically. I'm going to give the example of nicotinic acetylcholine receptors. So remember, nicotinic acetylcholine receptors is a massive, great family of loads and loads of cis-loop ligand-gated ion channels. Now, all of them are selective for sodium and potassium ions, okay, and they all have the same structures that make them selective for sodium and potassium ions, i.e. cations over anions. Okay, so I'm going to discuss what are these structural features that all nicotinic acetylcholine receptors have, which allows them to select for positively charged ions over negatively charged ions. Okay, well, let me just draw another picture then of the transmembrane domain here. Again, we'll need this picture of it in cross-section like so. So I'm just going to really just copy that picture out that I've got up there. Okay, so once again, here is that narrowing at the center, the hydrophobic girdle. So I'm still going to show it in the closed state. Okay, but we know now how it turns into the open state, which is that hydrophobic girdle is basically released, and all of the M2 helices therefore move outwards. Okay, right, so here is this cross-section through our cis-loop ligangated ion channel. I'll mark on the M2 helices again, so here in red, here are M2 helices. I'll mark on the hydrophobic girdle that is holding these M2 helices together there in that bundle and narrowing the pore at the centre to stop any ions moving through whilst you're in the closed state. But then once you're in the open state, how does the nicotinic acetylcholine receptor select for positively charged ions? Well, basically, it has 3 rings of positive charge. Oh, sorry, not rings of positive charge, completely wrong. Rings of negative charge that are around the pore, okay, and which therefore select for positively charged ions. So let me just show these on here. So in green here, this is basically a ring of negatively charged amino acid residues. So we have got the 5 M2 helices surrounding the pore here. Okay, and those M2 helices will have amino acid residues facing into the pore, and basically this green ring here is supposed to show a ring of negatively charged residues. Each one of the M2 helices, each one of the 5 M2 helices will have a negatively charged residue at this point, okay, and these negatively charged residues will be facing into the pore, okay, and that is now going to repel away negatively charged ions that would like to come into here. Okay, and it's going to be attracting in positively charged ions. So this is how the nicotinic acetylcholine receptors are going to select for positively charged ions. Now they don't just have one of these rings, they actually have 3 of them. Okay, 2 of them are on the sort of cytoplasmic side down here. There's one, and there's another one there. Okay, and these rings of negative charge are in fact given names. Okay, so the one on the extracellular side here, that's known as the extracellular ring, and I'll just abbreviate extracellular down to EC. Okay, the one in the middle, just below the hydrophobic girdle there, okay, that's called the intermediate ring. So this one is the intermediate ring. Okay. And then we've got the one that's underneath the intermediate ring that's on the cytoplasmic side of the nicotinic acetylcholine receptor transmembrane domain. Okay, and this then is called the cytoplasmic ring. Okay, so they're all rings of negatively charged residues. Okay, and the residues are in the M2 alpha helices. So it's a very similar concept to the hydrophobic girdle. Okay, but in the hydrophobic girdle, we had a ring of hydrophobic residues which were all interacting with one another and therefore bringing the M2 helices together, close together in the center there. Okay, in the case of these negatively charged rings, okay, it's exactly the same principle that you have on each of the M2 helices, a negatively charged residue, but their function is not to interact with one another. Of course, they're going to repel one another, not interact with one another. Their function is to select the positively charged ions to come through the pore and stop negatively charged ions coming through the pore. Okay, so that's believed to be how the nicotinic acetylcholine receptor selects for positively charged ions. Okay, so let me just give you then examples of negatively charged amino acid residues that could take part in these rings here. Okay, so aspartate and glutamate residues are the main 2 negatively charged residues. Okay, so let me just show these. So they have very similar structures. So I'll start by showing you aspartate. So here is the amino group here. Okay, here is the the alpha carbon with a hydrogen coming off it. Here is the carboxylic acid group here. Okay, now the R group of aspartate is then you just have a methylene group with then a carboxylic acid group coming off it. Okay, so here's the methylene group, here's the carboxylic acid group, and that carboxylic acid group will have lost its proton. Okay, it will have given it away to solution under normal physiological pH, and therefore will have a negative charge. Okay, so that's why aspartate residues have a negative charge. Okay, it's just like when we discussed glutamate previously. Glutamate was the conjugate base of glutamic acid. Aspartate is the conjugate base of aspartic acid, and once you've lost that proton away, you now have a negative charge, and you're no longer an acidic residue, instead you're actually a basic residue. Okay, glutamate then we've seen before, okay, but I haven't shown this as a residue before, but it's basically an identical structure to aspartate, except that now you'll have an additional methylene group before the carboxylic acid group, so you'll have two methylene groups there. So I'll just show glutamate now for completion. So here's the amino group, okay, here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group attached to the amino group of the next amino acid along, and the R group of glutamate consists of two methylene groups, like so, then with a carboxylic acid group off here, and again, that carboxylic acid group will usually have donated its proton away into free solution, and therefore will have a negative charge on it. Okay, so to summarize then, the way that the nicotinic acetylcholine receptor selects for positively charged ions is that throughout these M2 helices, you have points where you have conserved rings, basically, okay, where all of the M2 helices have a negatively charged residue, basically, okay, and this makes these rings of negative charge around the pore, okay, and this is going to select positively charged ions and repel away negatively charged ions. So that's how nicotinic acetylcholine receptors select for sodium and potassium ions over chloride anions. Okay, now there is a very similar concept to this in other cis-loop ligand-gated ion channels. So in other cis-loop ligand-gated ion channels, you have these rings of negative charge to select for positively charged ions, and then it inverts if you want to select for negatively charged ions. So if you want to make a chloride selective channel, obviously you need not to have negatively charged rings, but you need positively charged rings now, okay, to attract the negatively charged chloride in and repel away the sodium and potassium ions. Okay, now in other examples of cis-loop ligand-gated ion channels, you're not going to have the rings of charge in the same positions as you will have for the nicotinic acetylcholine receptor. So these free rings are specifically for the nicotinic acetylcholine receptor, but the concept generalizes basically. All cis-loop ligand-gated ion channels will have these rings of charge along the pore, basically, okay? And if you want to select for positively charged ions, they will have negatively charged residues in these rings, so aspartate and glutamate residues. If you want to select for negatively charged ions, then you need to have positively charged residues in those rings to create a positive charged ring. Okay, now let me just show you some examples of amino acids that have positive charge and therefore will be used in these rings of charge for the chloride selective ligand-gated ion channels in this cis-loop family. Okay, so these are the negatively charged ones. The two main positively charged amino acid residues are the amino acid arginine and also the amino acid lysine, but I'll firstly show you arginine. Okay, right, so again I'll just start by drawing the core amino acid residue structure here. So here's the amino group, here's the alpha carbon, here is the carboxylic acid group, and then the R group of arginine consists of 3 methylene groups, okay, which I'll just show like so. Here's one methylene group, I'll put it in brackets and then put 3 there. Okay, then after those 3 methylene groups, you then have an amino group, so nitrogen, the hydrogen coming off here, and then you have another carbon coming off this, and then off this final carbon here you have another amino group coming, and then a double bond to a nitrogen atom which is then also attached to a hydrogen. Now, this is the neutral arginine at present. However, arginine is a basic residue. It can accept a proton from free solution, and the reason it can accept a proton from free solution is that that nitrogen there that is double bound to the carbon, okay? And this nitrogen is very special. It has a very special name, and I'll just give you that special name. Okay, so this nitrogen that is double bound to the carbon, this is actually called the guanidino nitrogen. Okay, so you might see that referred to as the guanidino nitrogen. So basically the guanidinonitrogen of arginine has this lone pair of electrons, okay, because it only has 3 bonds at present, and therefore the other 2 electrons are in this lone pair of electrons. And now what can happen is that 3 protons from solution can come and associate with this lone pair of electrons here, okay, because that's the center of negative charge. Even though the molecule doesn't have a net negative charge, the lone pair of electrons is a center of negative charge, and therefore the proton has an electrostatic attraction to that lone pair of electrons. Now, of course, when you associate a proton onto the end of the arginine residue like this, that will then take the arginine residue from being neutral to being overall positively charged. Okay, so just like the aspartate and glutamate residues can lose their protons and become negatively charged, this basic residue, the arginine, can accept protons and become positively charged. Well, it's really the opposite, but that's how arginine can gain a positive charge. And we're going to see a very similar thing for lysine. Okay, right. So let's now talk about lysine then. So again, I'll start by just drawing the core amino acid structures. So here's the amino group here. Okay, then you've got the alpha carbon with a hydrogen coming off it. Then you've got the carboxylic acid group down here, which will be linked to the amino group of the next amino acid along. Okay, and the R group of lysine then consists of 4 methylene groups. So I'll show these here. So here is 1 methylene group, and you'll repeat that 4 times. And then right on the end of those 4 methylene groups, you'll then have an amino group, like so. Okay, and again, that nitrogen of this amino group will have a lone pair of electrons, and what can happen is a proton can come and associate onto that lone pair of electrons of the nitrogen on this neutral lysine residue here. Okay, and that can then give the entire lysine residue a positive charge. Okay, so the running theme here is that the actual normal residue is uncharged, okay, but either by losing its proton or by gaining a proton from free solution, it can become charged, basically, okay, and we've seen that the aspartate and glutamate residues are the conjugate bases of the acid molecules, which then have the negative charges, okay, and arginine and lysine can become protonated to gain positive charges. So basically, in these anion selective cis-loop ligandated ion channels, you are going to have rings of arginine and lysine residues which can become protonated under normal physiological pH, and therefore you'll end up with rings of positive charge around the pore, and these rings of positive charge are therefore going to repel away the positively charged ions and attract in the negatively charged chloride anions. Okay, and that's how you can achieve chloride selectivity. Okay, right, so that concludes our discussion of the transmembrane domain. We've now seen the hydrophobic girdle, which is the gate of the channel. That's what determines whether the channel is open or not. When the hydrophobic girdle is assembled, it's closed. If you break the hydrophobic girdle, the M2 helices spring back outwards, okay, and that opens the channel. Okay, we've also seen what determines selectivity for either cations or anions, and that's these rings of positive or negative charge. Okay, right. So in the next video, what we'll do is we'll turn our attention onto the intracellular domain, which is only present in the mammalian cis-loop ligandated ion channels, and then we'll go on to the extracellular domain, and then finally we'll turn our attention onto the gating mechanism.